Hello everyone, I'm Charlotte from MDPI and I will be your host for today. Welcome to the second live session of the first International Electronic Conference on Toxins, focusing on mycotoxins, their involvement in fungal plant infection, animal-human toxicity, and potential therapeutic applications. This e-conference is hosted online from the 16th to the 31st of January 2021 and is organised by MDPI and the journal Toxins. I would like to thank the conference chairs, the conference committee and the session chairs for making this e-conference possible, along with the speakers and of course the attendees. The conference papers and presentations are available on the conference webpage for discussion during the time of the conference and will be published in the journal Proceedings. The 4 live sessions of this e-conference will also be made available on our conference webpage. I would like to remind everyone that this session is recorded and the recording will be made available as soon as possible and will be posted online. This webinar is also being live streamed on MDPI's YouTube channel, so hello to those of us watching on there. Today's live session is chaired by both Dr. Laura Soler Vasco and Dr. Veronika Nagl. Born in Spain, Dr. Soler Vasco is Doctor in Veterinary Medicine by the University of Murcia, Spain, and holds postdoc at KU Leuven in Belgium and INRAE Val de Loire, France, on pig intestinal pathophysiology and clinical proteomics. Since 2017, she is a junior researcher at the Biosynthesis and Toxicity of Mycotoxins group from INRAE Toulouse and is interested in applying systems biology approaches to reveal the toxicity of mycotoxins. Dr. Veronika Nagl works as scientist at the BIOMIN Research Center in Toulouse in Austria. Veronika studied veterinary medicine at the University of Veterinary Medicine in Vienna. Thereafter, she did her PhD in the Christian Doppler Laboratory for mycotoxins metabolism at the University of Natural Resources and Life Sciences, also in Vienna. Her research focused on the development of LC-MS/MS-based biomarker methods to elucidate the metabolism of masked mycotoxins in rats and pigs. After a post-hoc period in the same lab, she joined the BIOMIN Research Center in Toulon in Austria in 2014. There, her main research topic is the investigation of molecular effects of Fusarium mycotoxins in livestock species. Special emphasis is put on employing omics technologies and utilizations of results for biomarker development. So far, Veronika has authored and co-authored more than 20 articles, book chapters, and patents, and presented her research at multiple international conferences. Thank you to both of you for chairing today's session. Each presentation will be followed by a question time. If you have any questions, please feel free to post them in the Q&A box at the bottom of your screen. If you wish to ask your question in person, please raise your hand and I will give the microphone. That's it from me. I now hand the floor over to our 2 session chairs and wish everyone a nice webinar.
Thanks, Charlotte, and hi everybody from my side. Without any further ado, I will introduce the first speaker of the day, who is Dr. Isabelle Oswald. Dr. Isabelle Oswald is the head of the INRAE Research Center in Food Toxicology at Toxalim in Toulouse in France. In this research center, she leads a research team of 20 persons, and with her team, she is focusing on the one hand on characterization of the production of mycotoxins and other secondary metabolites of fungi, and on the other hand, to determine the toxic effects of mycotoxins in rodents and pigs using different models from in vitro, ex vivo to in vivo. Currently, Dr. Oswald and her team is focusing on the effects of emerging mycotoxins and toxin mixtures. Isabelle has more than 200 international publications and is one of the most highly cited authors in this field. She's an expert for different institutions like EFSA, ANSES, and CHECFA. And in 2018, she received a Lifetime Achievement Award from INRAE. And this January, she was appointed as Knight of the French Legion of Honor. So, félicitations à la Reine Française de Mycotoxines. Isabelle, the screen is yours.
Thank you, Veronika. And here, during this presentation, I'm going to talk about the evaluation of mycotoxin and especially about the risk assessment of this mycotoxin and more from the European side. So, in the framework of risk analysis, there's three pillars. The first one is risk assessment and is done at the European level by EFSA, European Food Safety Authority. Then the second pillar is the risk management, and in Europe is done by the European Commission. And then there's also the risk communication, which is an interactive exchange of information and opinion. So now we are going to focus more on risk assessment. The risk analysis is made of three pillars: the risk assessment, the risk management, and risk communication. Now we are going more to focus on risk assessment. So the risk assessment can be divided in several parts. First, there is the hazard identification. Then the hazard characterization that allows to determine a tolerable daily intake. And then when we compare the hazard characterization to the exposure assessment, this leads to the risk characterization. So now we are going to go step by step. First, the hazard characterization and the establishment of a tolerable daily intake. The hazard characterization is made by identifying. In case of mycotoxin, we go through all the literature and we determine what are the very important effects and what are their relevance for human. Then we identified no observed adverse effect level for each critical studies and for each critical effect and for each studies. Then there is an identification of the pivotal studies, and usually this is the studies which has the lowest NOAEL. Then for human, we applied a safety factor to the NOAEL to establish the tolerable daily intake. The default safety factor is 100, but it can vary, as we'll see. For animal, what is very different for human, there is no safety factor that is applied to the NOAEL to define the tolerable daily intake. So this can be summarized in this scheme here. We have a dose effect response, then which allows the determination of the no observed adverse effect level. And then with the uncertainty factor that is applied for human, you define a tolerable daily intake. How is calculate the safety factor? So this safety factor, as I already told, is a factor by which the NOAEL will be multiplied to obtain a TDI for human. So this factor is composed first of a factor 10 to take into consideration the intra-species variability. Indeed, we consider that human is 10 times more susceptible than the most susceptible species that we have determined in the literature review. Then there's another factor 10 to take into consideration the intervariability between human. It doesn't take into account the variability between adults and infant or adults and elderly people. It just takes the variability between normal adults. Then there's another factor from 1 to 50 to take into account the different effect. If we have a non-very serious effect, for example, for Deoxynivalenol, we have the decreased feed intake, we'll have a factor of 1. But for some other molecule, we have a factor of 50 when the effect is more serious. This is just an example of that for zearalenone. Zearalenone, we know that it's an estrogenic molecule, and the female piglet is the most sensitive species. In this species, the NOAEL is 10 microgram per kilogram body weight per day. Then we need to apply the safety factor. But we realize that toxicodynamic information indicate that it's likely that the human female would not be more sensitive than the pig female. So for the derivation of the TDI, it's not necessary to include this toxicodynamic information, because I didn't told you, but this 10 other factor is in fact 2.5 multiplied by 4. So here we don't need to have this 2.5 for the toxicodynamic, but we keep the 4 for the toxicocinetic. So we then have a safety factor of 40, 10 times 4. And then the TDI for zearalenone in human is 0.25 microgram per kilogram body weight per day. This is here the example for animal. As I told you, for animal, there is no safety factor. So we just consider the lowest NOAEL or lowest LOAEL. Here is the lowest observed adverse effect, and here's a no observed adverse effect level. So depending on the experiment, unfortunately, sometimes we don't have a NOAEL, so we just have a LOAEL. And this is those values that are used to determine the toxical value for animal. And they can be expressed as, usually in animal trial, it's expressed in kilogram feed. So then it's translated in kilogram body weight per day. There is another way. Now it's more and more a new concept that has emerged in risk assessment, is not to use the NOAEL, but to use the benchmark dose. Why does it happen? It's just because when I show you an example using the NOAEL, you see that from an experiment, you're using only one point, the point where there's no effect. The concept of benchmark dose is instead to use all the dose response. And from this dose response, basically, you determine a NOAEL, which is a benchmark response. But the concept of the benchmark dose is really not to use just one point, but to model the dose response and then to derive the effect from that. And usually we use the BMDL10, which is a 95% lower confidence limit of the benchmark dose for 10% extra risk of the critical effect. This is an example where this benchmark dose has been used in mycotoxin, and this is the example of the T2 toxin. So there is an subchronic toxicity study in rat, a 90-day subchronic toxicity studies, and the pivotal effect is a dose-dependent reduction in total leukocyte count. With using this BMDL approach, we have a BMDL10 of 3.3 microgram T2 toxin per kilogram body weight per day. Here we had an uncertainty factor of 200, 10 for the interspecies, 10 for the intraspecies, and 2 because it was a subchronic toxicity study, not a chronic toxicity study, only 90 days, and the toxic effect progressed during the whole period, never reaching a plateau at the end of the study. So we extrapolate that if you reach a plateau, it can be, we had a safety factor of 2. So with that, the TDI that was derived was of 0.02 microgram T2 toxin or HT2 toxin per kilogram body weight and per day. In fact, it was wrong from the real number, which was 0.017. The concept of benchmark dose you can easily understand is how are you going to modelize the data. And you see, this is 2 modelization. Depending on the modelization, you don't have the same response. So now, especially at the European level, there's real guidance how to modelize which model you can use. And when several models are possible, now usually we use the mean of the different models that are considered okay for derivation of a benchmark dose. So now we've seen what happened is hazard characterization and establishment of the tolerable daily intake. Now this has to be looked in regard to the exposure assessment. The exposure assessment is quite easy to understand. So this is the dietary exposure is the consumption multiplied by the concentration of the food chemical divided by the body weight. So the consumption data are data on the level of consumption of different food in milligram or microgram per kilogram body weight per day. And we have table both for animal and for human. And for human, it also depends on the region of the world, because you all know that you don't have the same diet. And for example, at the level, European people doesn't have the same diet as an African people or a people from Asia. In Europe, there is only one diet that is considered, but when you have evaluation from the GEFCA, so a worldwide evaluation at GEFCA, we have different diet depending on where you live. And then the food chemical concentration. The food chemical concentration is a concentration of the mycotoxin in the different compound that compose the diet. And here also, when you are, for example, when you have an evaluation at the international level, the food chemical concentration will depend on where the chemical has been analyzed. You don't have the same level of concentration in the different part of the world. So, then to the risk characterization is really to compare the exposure and the tolerable daily intake. And in fact, it's pretty easy. There's two situations. Either the exposure is below the TDI, so there's no risk for the consumer, whatever it's human or animal, or the exposure is above the TDI, and then there's a risk for consumer, human or animal. So here is here's an example. The risk characterization of DON and DON derivative in human. And for DON and modified form, the group TDI has been established of 1 microgram per kilogram body weight per day. Then, if you look at the estimated chronic dietary exposure and you compare it to this TDI, this is a table that has been done at the European level in the different group. So if you use a mean dietary exposure, you see that some groups, the toddler or the children, at the lower bound, but at the middle bound even more. And when you consider not the mean dietary exposure, but the people that are highly exposed, the 95 percentile dietary exposure in the total population, here you see that quite a lot of number are above 1. So it indicates a potential health concern considering the exposure of human to DON and modified form. This is a risk characterization that has been established here for zearalenone for animal. And here you see that whatever the species, sheep, gilt, piglet, chicken, turkey, fish, or dog, when you compare the estimated, the exposure and the NOEL or the LOEL, here we are always below 1. The highest will be here 21% of the NOEL or the LOEL. So the risk of adverse health effect of ZEN for animal is considered extremely low for poultry and low for sheep, dog, pig and fish. Here, what I talked about before was all the mycotoxin except the one that have a genotoxic or a carcinogenic activity. Indeed, for this compound, the GEFCA say that they need to be present at irreducible level. It means as low as possible. And other organizations refer to that as ALARA, as low as reasonably achievable. So basically, it indicates that for those compounds, you don't want to have any of these, you don't want to have this mycotoxin in your diet. But anyway, having said that, it's important to evaluate and to classify the mycotoxin, and this is possible using the margin of exposure. So what is the margin of exposure? Is a tool to consider the possible safety concern arising from the presence in food or feed of substances that are both genotoxic and carcinogenic. So the margin of exposure is a ratio between two factors, which has, and these two factors are the adverse effect and the level of exposure. If the margin of exposure is below 10,000, there is a concern. If the margin of exposure is above 10,000, it considers that this substance is of low concern. This is, for example, the case for aflatoxin. We know that aflatoxin is a genotoxic and can cause hepatocellular carcinoma in humans. So it's both genotoxic and cancerogenic. The pivotal study is the incidence of hepatic cancer in male rat following aflatoxin exposure. And using the benchmark dose, the limit is 0.04 microgram per kilogram body weight per day. And when we calculate the margin of exposure for aflatoxin for the different group, you see that here it's only 5,000, and sometimes in different situations it can be quite low as 44 or around 1,000. So really that indicates that the margin of exposure for aflatoxin range from almost 1,000 to 28 for the highly exposed people across the different survey and age group. So it's below 10,000, so it indicates that really aflatoxin is of health concern, even in the European population. Here, this is the general case I talk about, but sometimes there is insufficient toxicological record, or it's impossible to determine the TDI. Then it's important how can we characterize the right risk without having characterizing the danger. So here, in this case, we can use margin of safety, threshold of toxicological concern, read across, weight of evidence of mode of action. So this is new approach that are used when we have really not much data. So here I really present you what about risk assessment, but it's also important that when risk assessment is done by scientists, then it's a role of risk management to assess policy alternative and to select and implement the appropriate option. And during all this process, risk communication need to be done. So now I want to finish my presentation by talking about the new challenge in risk analysis of food contaminant and especially of mycotoxin. And I think there is two issues that for me are very important, is the non-monotonic curve and the mycotoxin mixture. So the non-monotonic curve are those type of curves that go which are not that intuitive because we all have in mind that the dose make the poison, as Paracels say, but it's not always the case. In some case, you have a response at a dose and no response at a smaller dose or not response at a higher dose, but you also have, like for example, for the bisphenol, you have a response at a low dose, then no response again. So it's very difficult to assess, to have a proper risk assessment in this case. Hopefully, in the case of mycotoxin, no example have been described in this case. The other problem is a problem of mycotoxin co-contamination. We know that we are exposed simultaneously to several mycotoxins because fungi produce several mycotoxins simultaneously, because food might be contaminated by several fungi, and because meals are composed of multiple raw material. So we know this is just an example from a BIOMIN survey that look at samples that were contaminated. In this survey, we have 19% of the sample that were contaminated below the detection level, 33 with one mycotoxin, but the majority with more than one mycotoxin. So co-contamination by several mycotoxins is the rule, not the exception. The additive effect is in fact taken into account so far by EFSA. EFSA opinion, there's several EFSA opinion on group of mycotoxin. It concerns aflatoxin, fumonisin, and most mycotoxin and their modified form. And for that, they determine toxicological equivalent factor, and the risk assessment is made on this toxicological equivalent factor, then considering an additive effect. This is, for example, the case for zearalenone and its modified form. If we consider zearalenone with its estrogenic effect, it can be considered, if it's considered as one, there are some modified form of zearalenone that are less toxic than zearalenone, but there are also modified forms that are more toxic than zearalenone with a factor of 60. So what EFSA suggests is to sum up all these different mycotoxins in the food or in the feed, and applying to each contaminant the safety factor the toxicological equivalent factor that varies from 0.2 to 60. But we add that, so we have the analysis of the sum of Zen and modified form. And in this, so we consider additive effect, but this, So this example shows you that we can consider mycotoxin mixture, but it's only considered as additive effect, taking into account their potentialization. But synergy and antagonistic effect are not taken into account. And we and others have, for example, demonstrated that synergistic effect occur within mycotoxin. We have demonstrated in both human cell and porcine cell that, for example, DON and 3A-DON have a synergistic effect. And this was done at low concentration. And we conclude that at low concentration, synergy is the main type of interaction that was observed between trichoticin. This experiment were done either in vitro or ex vivo in explant level. So synergistic effect and antagonistic effect are not taken into account. We can wonder why. I think it's difficult because due to the multiplicity of the situation. And in addition to what I showed you before, we also demonstrate that the type of interaction can vary at different concentration with the same mycotoxin. So two mycotoxins at a higher concentration can have an additive effect, and at a lower concentration can have a synergistic effect. So it's really a Pandora box to determine how the synergistic and antagonist effect can occur. And it will require to integrate in vitro toxicological testing in risk assessment, which is not the case so far. So the take-off message of this presentation: risk analysis include risk assessment done by EFSA, risk management done by the European Commission at the European level, risk communication. The risk assessment include hazard identification and characterization. This leads to the establishment of health-based guidance value. Then, when we compare with the exposure assessment, we are able to determine the risk characterization, which is a comparison between the health-based guidance value and the exposure. Mycotoxins are natural contaminants for which no regulatory dossier are needed. So the toxicological data that we have are scarce. For example, compared to pesticide, the pesticide dossier are much more, there are much more experiments included in this dossier. And this is especially true for new mycotoxin, emerging one or modified form, for which the number of data are really low. The risk assessment should also take into account this new mycotoxin. We didn't talk because it was not the topic, but climate change, which will change mycotoxin exposure. And we talk about it, mycotoxin mixture. I want to thank my colleague in Toxalim and also you for your attention. Thank you.
Thanks, Isabelle, for this very nice overview on the topic of mycotoxin evaluation and regulation. We've already received a first question via the Q&A chat. Maxence, I hope I pronounced you correctly. Do you want to ask the question yourself? Otherwise, I can read it out.
Yes, I was saying thank you very much for your presentation, really clear, and it was a really nice presentation. I have a small question regarding the BMDL model. In some cases, I noticed that the opinions are using a parametric bootstrap concept, and I don't know how it is applied and why with the same data set it is possible to find different BMDLs.
It's going to be difficult for me to answer because if I know the theory of BMDL, I don't use that practically. There's two things in your response. I think we use bootstrap to increase the number of occurrence. So this is a way to have more data and to generate more data. That's one question. Why do we have different BMDL? This can be also depending on the modeling that we use. And that's something that is more and more important. And before I would say 10 years ago, when they were using BMDL, they were using only one model to feed the data. And the problem is, especially when having mycotoxin, we don't have a very important dose effect. We don't have that much data to have the dose response curve. So it's important to analyze different models to try to mimic the best. And usually now what they are doing is they are using different models. For each model, they are defined parameters saying that it's okay and it's not okay. And all the models that are okay then are summed up to have the BMDL. I don't know if I answer correctly the question.
Yes, okay. Thank you.
There is another question from Marc. So Marc, please go ahead.
Nice presentation, Isabelle. I agree with you that normally the mixture of mycotoxin, the animal models, and what we have in the literature show that is not at risk. But what about mixing mycotoxin not only with other chemicals, but also with other factors that can be causing injury to the animals, such as, for example, bacteria or viruses? Because I mean, Siska works on this, and we did also, and you did also, and we show many groups shown that some mycotoxin can increase a lot the infection process. Or so, is it any way, or is it any study in vivo about this?
There are studies in vivo. There are more and more studies in vivo showing that mycotoxin exposure can change the susceptibility to mycotoxin. We'll have studies we'll have an example at the end of this session showing that mycotoxin can increase inflammatory bowel disease. So that's a fact. Now the question is, how can we integrate that in the risk assessment? And then this is a problem. How should we, so far, the risk assessment is not made by a specific group unless vegan. But we can imagine, for example, later having more specific group, for example, groups suffering for one pathology and knowing, but we need to have more data on that to be able to do that properly. And so far we don't have it even in animal model.
Let's continue with our second speaker, and I will hand over to Laura for that.
Hello, I have the pleasure to introduce our next speaker, Siska Croubels, which is a graduated as Master in Pharmaceutical Science from Ghent University. She obtained her PhD in the field of bioanalysis of veterinary drugs. And she was a postdoc at the Faculty of Veterinary Medicine from the UU Ghent. Then in 2004, she was appointed as an assistant professor of veterinary toxicology at the same faculty. And since 2014, she's a full professor and director of the Department of Pharmacology, Toxicology, and Biochemistry. Her research in the field of veterinary toxicology focuses on the toxicokinetics and toxicokinetic modeling of mycotoxins, on the interactions between mycotoxins and pathogens in several animal species, and on the development of in vivo and in vitro models for efficacy and safety testing of mycotoxin detoxifiers. Her research group is a member of the Mytox and Mytox South Association research platforms that are dealing with the effects of mycotoxins on human and animal health in the North and South. Siska, it's up to you.
Yes, hello everybody. I will share the screen. So thanks a lot for this nice introduction. And also I want to thank the organizers and also Marc for inviting me to this webinar. Today I want to talk to you about the fate of major mycotoxins in pigs and poultry, and also in growing piglets as a kind of surrogate animal model for children. Actually, this presentation perfectly aligns with the presentation of Dr. Oswald, where Isabelle just talked about risk assessment. And that's the part of the general risk analysis together with the communication and the management. And Isabelle showed that the risk can be characterized based on these three pillars, the identification, the characterization, and the exposure assessment. I have to say, within scientific community, a lot of research is being performed on these exposure assessments, and many researchers determine the occurrence of hundreds of mycotoxin and metabolites in several food and feed matrices. And when we combine this with the consumption, then you can actually determine the external exposure, or let's say the dose of mycotoxin for a human or an animal. Far less research is being performed on the toxicodynamics and kinetics. And today I want to highlight some aspects of these toxicokinetics, because these processes really determine the internal exposure or the internal dose that can arrive or be present in animal and human beings. Okay, just a quick overview. What's the difference between TD and TK? The toxicodynamics study the effect of the toxin on a body, so the toxicity, based on suitable in vitro, ex vivo, in vivo models, and the mode of action is there analyzed. Whereas the kinetics really study the effect of the body on the toxin, and it comprises 4 major processes: the absorption, the distribution, the metabolism, and the excretion. So together, these are the 4 major ADME processes. And for each of these 4 processes, the rate and extent of the process really is dependent on a lot of factors. For instance, the absorption is dependent on the pH of the stomach or the 4 stomachs in ruminants, on the gastric emptying time, the motility, surface area, presence of enzymes in the lumen or in the gut wall, and eventually also presence of protein transporters to which mycotoxins might be substrate. So all these together determine the oral bioavailability, or F%. That's by definition the percentage of the dose that is absorbed and enters the systemic circulation in its unchanged form, so as the parent mycotoxin. Next, the distribution takes place, and it's dependent on the plasma and tissue protein binding of the toxins. Also on the body composition, I'm thinking of water-fat ratio, of specific barriers in the body, and of organ blood flows. The TK parameter there is the volume of distribution, and it's a measure of the extent of the distribution of a toxin in the body. Next, the metabolism mainly takes place in the liver, but also other organs can metabolize, like the intestines. And there we have the processes of phase 1 and phase 2 biotransformation reactions. Amongst phase 1, the cytochrome P450, or an important enzyme family responsible for oxidation. Also, ABC transporters, these active protein transporters, can play a role in the metabolism by effluxing the mycotoxins, by eliminating them. And then finally, the renal excretion is dependent on the filtration, eventually active tubular secretion, and also eventual reabsorption from the primary urine into the blood. So both metabolism and excretion represent the elimination of the mycotoxin, determined by this main TK parameter, total body clearance, actually defined as the volume of plasma that is cleared from mycotoxin per time unit. So distribution, volume of distribution, and total body clearance are the primary toxicokinetic parameters. To determine these, we need specific toxicokinetic trials. But a golden standard, let's say, of such trial is a crossover study design, where actually a group of 6 to 8 animals are divided in 2 groups, where the first group receives a single intravenous bolus dose of the mycotoxin, and the other group receives an oral dose of this toxin. And after a suitable washout period, usually taking into account at least 7 times the elimination half-life, then the groups are switched. So each time after each of the 2 administration routes, repetitive blood samples are collected and plasma concentrations are determined using targeted LC-MS methods and also untargeted high-resolution mass spectrometry to map the whole metabolism in the body. Then the plasma concentration time curves are constructed, and the absolute oral bioavailability F% is calculated by taking the ratio of the AUCs of the oral dose compared to the intravenous route. And then finally, suitable kinetic modeling is performed, usually using dedicated software. This can be done by the conventional compartmental modeling, where the body is theoretically divided in compartments, like this central square here, central compartment, where the blood belongs to. And for some mycotoxin and some species, you can have second peripheral compartments or a third compartment. And each time the movement of the toxin between these compartments is quantified by mathematical formulas. Sometimes it's needed to build customized, tailor-made models, like this is the case here for DON3-glucoside, to really map what happens in the body with this mycotoxin after intravenous dosing and after oral dosing. And again, you see these compartments which are interconnected to each other. Okay, so when performing these kinetic studies, this can be done in many relevant animal species. And this gives us information to be able to compare these processes, these ADME processes, and finds similarities between species or differences. And then it's very nice to try to explain if differences are seen, why this happens. But when we look to the pig can also be used as a large animal model for human beings. Like Viviane yesterday mentioned in her talk, as the pig is known as a suitable adult animal model for humans due to really striking similarities in physiology, in anatomy of major organs involved in ADME, like the cardiovascular system, the GI tract, and the liver and kidneys. Okay, but what about children? Children have usually a higher vulnerability concerning mycotoxins simply because they have a higher external exposure. They have a higher food intake, scaled for body weight, and are really high consumers of cereal-based products. But children can also have a higher internal exposure due to immature, to metabolic and physiological immaturity, let's say, of major organs involved in these kinetics. So the question can be raised, can we use piglets and growing piglets as a suitable model to study kinetics in children? So indeed, major developmental changes occur during growth of a child and also of a pig. And when performing such type of studies, it's good to make categories, age categories, and like shown in this table, we have here 4 age groups from neonate to adolescent. And the big advantage of doing research in pigs is that they reach puberty, let's say within the time frame of 6 to 7 months, depending on the sex. We characterized some parts of the ADME processes in these growing piglets, and we could conclude that it might be suitable infant and child model, for instance, based on the ontogeny or let's say the ripening of, let's for instance give the example of cytochrome P450 enzymes. We could map their expression by proteomics, so the enzyme abundancy and also their activity in piglets of several age categories. And we compared it with children. The same we did with the glomerular filtration rate to see what are the difference or comparisons between pigs and humans. So it's important to have then suitable tools, of course, to perform such intensive in vivo trials. For instance, here for blood collection, we can easily catheterize the jugular vein to take systemic blood. But it is also possible to take blood from this vein here, the portal vein. This is done through catheterization, and this medical image here shows you the course of this catheter. And then they are both tunneled upside here so that we can easily, we have here two ways, easily collect both systemic blood and pre-systemic blood to study what happens with mycotoxins or other xenobiotics in the gut or in the gut wall, for instance, if we are able to collect blood of these portal veins. Then urine can also be collected and feces. But for urine, even a non-invasive tool is available, so no catheterization is needed. By simply sticking urine collection bags, plastic bags, to the belly of only male piglets, it's possible. So the aim of today, further on, I want to show you some examples of these comparative toxicokinetics of some emerging and modified fusarium toxins in both pigs and poultry, and also in the growing piglets as a model for children. Knowledge of these toxicokinetics can give us insight in possible species-dependent differences in sensitivity between animal species. Also, within one animal species, it can give us information on difference according to the age of the animal. And performing or studying the kinetics can give us also information on suitable biomarkers for each species to determine exposure to mycotoxins. So more for the diagnostic part, to diagnose mycotoxicosis. or these biomarkers can also be used for in vivo efficacy testing of novel candidate mycotoxin detoxifiers. So I will show you now some examples of DON and its modified form DON glucosides, also Zen and some inyatins. Let's start with DON. This gives you an overview, a summary of the main toxicokinetic parameters in 2 species, pigs and broiler chickens. And one can easily see some remarkable differences. For instance, the oral bioavailability F percentage is only 5% of the dose that enters the systemic circulation in its unchanged form in a broiler, compared to 80% in a pig. Also, a clear difference in the clearance is seen. 88 milliliter per minute is cleared in a broiler, and only 5 milliliter per time unit is cleared in the pig. The volume of distribution is somewhat similar, a little bit higher in the broiler, and this significantly higher total body clearance results actually in a significantly shorter elimination half-life, only 18 minutes to halve the plasma concentration in a broiler compared to 160 minutes in a pig. So taken together, this lower bioavailability and higher clearance results in reduced systemic exposure in the broilers. And that's why broiler chickens are more resistant to the effects of DON compared to pigs, at least where these are more sensitive species. Let's look now to the metabolites of DON, phase 2 metabolism. In pigs, it's known, also in humans, that DON glucuronides are formed and are nicely formed here, the orange curve, after a single oral dose. And in broiler chickens, we could not, or could detect only traces of DON glucuronides, but especially there, almost no DON was present anymore, this high clearance, and quite easily this DON sulfate metabolite was formed, another phase 2 conjugation reaction. And if you look to the y-axis, we have here dealing with the same dose per kilogram body weight. It must be concluded that the broiler chicken has a kind of extensive sulfation, easy pathway, and the pig also, but it's more moderate compared to the chicken. So that might be a second explanation why broiler chickens are more resistant. They easily form a non-toxic water-soluble metabolite compared to the pigs. We did similar research in different poultry species. I'm showing here the case of turkeys. The graphs show you plasma concentration time profiles again of DON in blue and then in gray, the DON sulfate metabolite. Both form this metabolite. And if you look to the y-axis, it's a log scale. It seems that broilers form it more easily than turkeys. So if you then compare the ratio of DON divided by DON sulfate, it's a higher ratio. It means that turkeys have a kind of moderate sulfation compared to chickens having a more extensive, easy sulfation. So that again gives an explanation why turkeys might be more sensitive to DON compared to broiler chickens. Okay, then I want to show you the case of DON glucosides, where we performed some kinetic studies in growing piglets of different age categories to see if there are possible age-related differences. So first we did it in the youngest age category of piglets of only 4 weeks old, just weaned piglets starting to eat their cereal-based diet. The graph shows you plasma concentration time profile again, and the blue lines gives you the DON concentration after DON glucoside was administered. So we only administered DON 3G, but apparently DON is formed in the body, and both DON in the systemic circulation as in the portal vein are shown. And then the pink curves gives you the profiles of the metabolites, the 2 glucuronides. Here some nice conclusions or interesting conclusions can be seen or can be taken. First of all, we were not able to measure any DON 3G the parent toxin, in plasma. So only DON. That would mean that almost a complete hydrolysis of DON3G to DON takes place in vivo. Also, no DON3G could not be measured in the portal plasma. So that would mean actually that the hydrolysis takes place already in the gastrointestinal tract. And if you then have a close look to the first time points here, the beginning of the curve, we actually see a lag time of really about 3 hours before plasma concentrations of DON could be measured. This lag time indicates that this hydrolysis takes place more in the distal parts of the gastrointestinal tract, probably by a colon microbiota. And then the last conclusion is the metabolites are present, but in a low concentration, so relatively low phase 2 biotransformation reactions take place here. If we then translate these graph data to the really figures, the quantitative data, this table compares the kinetic parameters in 4-week-old pigs, so our young ones, to 11-week-old pigs. A similar dose based on milligram per kilo body weight was given. I will go with you through the table. The first one we see here is the total body clearance, and it's really significantly lower, only 5 milliliter per minute in these weaned piglets compared to a higher clearance in the more older pigs. This could be expected based on the lower biotransformation pathways and the probably lower renal clearance. The second conclusion is on the volume of distribution. This was higher in the weaned piglets compared to the older ones, probably related with the body composition. In the younger ones, we have a higher body water fat ratio. So a compound like DON with a low D value, rather low, it's more polar. It's logic that it's distributing more in the water compartment. So a higher volume of distribution there. And also we determined plasma protein binding, which was neglectable, was not present in plasma. Then the elimination half-life was really longer. So a longer presence of DON here after DON-3-glucoside administration in these younger piglets due to a combination of two things, the lower clearance and the higher volume of distribution. So 6 hours compared to only half an hour. Then looking to the absorption, it's expressed here as FRAC percentage, that's the percentage of the dose that is absorbed, both as DON and as DON-3-glucoside. It was higher, also Cmax was higher in the weaned piglets, probably due, but we could not definitely demonstrate that for sure, but to a higher intestinal permeability, more chance of a leaky gut at this age of 4 weeks, and the longer transit time. So longer action of enzymes could take place to cleave also DON out of DON-3-glucoside. And this was also reflected in the longer lag time, this longer transit time in this young age category. So really significant differences between both ages. And to summarize this, we can conclude that in the young age category, there is a higher internal, the lower clearance, higher volume of distribution, but also higher intestinal exposure to DON and DON3G in just weaned piglets. It can possibly be assigned to this leaky gut phenomenon or stress. This is the just weaning period with some barrier damage, let's say, and the higher water-fat body ratio, the longer GI transit time, and the lower phase 2 biotransformation capacity due to more immature liver. Okay, then the case of zearalenone, there it's important to look to the phase 1 metabolism because we have the alpha cell and beta cell, especially alpha, which has a higher binding affinity to the estradiol receptor, so more responsible for these estrogenic effects. We did kinetic studies in 3 poultry species, a broiler, a layer, and a turkey. The curves show you, the blue ones is the Zen profile, and then orange and gray, the alpha and beta cell metabolites. You can easily see that all three species form both alpha and beta. But the turkey is here again quite exceptional. It forms more alpha cell. It approaches the amounts of the parent compound here compared to the broiler and the layer. So taking into account this binding affinity, the potency of alpha cell, turkeys might be more sensitive than the other. So even within one bird species, one would say there are these differences based on differences in biotransformation. Then a final example is on eniatins, more emerging mycotoxins for non-regulated so far for animal feed. We have here kinetics in the pig, where we administered the 4 eniatins separately, A, A1, B, and B1. And even after the single dose, you can see clear differences, the higher profiles, higher Cmax, higher area under the curve, so higher exposure of eniatin B and B1 in pigs compared to the A and the A1. So the oral bioavailability will be different between these 4 compounds. A similar study in broiler chickens, where we administered B1, we had sufficient toxin available to dose 6 broilers, both intravenous route, this is the highest curve, and then the lower curve, that's the oral administration, the parallel elimination, but with more standard deviation can be seen, and especially the lower AUC. So remind, this is log scale. That means that you have really a low area under the curve, which is a measure for this systemic exposure. So again, translation to the kinetic parameters. Actually, we can conclude that we see more or less a similar trend as for DON. Between chickens and pigs, we have the low oral bioavailability. Only 5% of the dose reaches unchanged to the systemic circulation compared to 90 or 91% in pigs. That's almost complete absorption. And the broiler also has a higher body clearance, more efficient in eliminating the toxin, 6 liters per hour compared to only 2 liters per hour in the pigs. Here, the volume of distribution is also higher in the broiler, leading then to a higher elimination half-life. But in general, one can conclude, especially looking at this only 5% F and this high clearance, that again, the broiler chicken might be more resistant to eniatin B1 compared to a pig in this case. Okay, some conclusions, our future perspectives here. So if you want to study the kinetics, it's really important to do species-specific studies and not extrapolate just from a broiler to a layer or to a turkey or others, or between other species. And these kinetic studies can give you insights on possible differences in toxicity. And can you explain something there? It gives also insight in some suitable markers that are usually phase 1 or phase 2 metabolites like the glucuronides that are quite easily formed in some species, and that can be suitable compound to follow if you want to demonstrate exposure to mycotoxins or if you want to test efficacy in vivo of a candidate detoxifier. Next, we demonstrated age-related differences in internal exposure to modified toxins. I showed you the example of DON in the growing piglets. We did similar work with Zen in growing piglets, and the conclusion was quite similar. The infant age category is more exposed to than the older child category. And then one last conclusion, just from a practical point of view, to perform such kinetic trials, one need, of course, sufficient quantity of purified mycotoxin to perform these studies. And in this field of research, it's quite can be quite hampering factor and quite really an expensive thing compared to drugs like antibiotics, which are more easily available, of course, and cheaper. So what's the way forward? What's next and future? I think it's really time to move to other modeling approaches besides this in vivo kinetic modeling, and even skip it and go to in silico modeling to predict plasma concentrations. I'll show you final 2 slides. Here, population toxicokinetic modeling, which is more elaborate than the conventional compartmental modeling. But this is really a prediction of the whole population of species. And the big advantage is that it takes into account a lot of covariables like age, like body weight, gender. This can be included in the model, this POP-TK modeling. And then if we abandon in vivo and go to in silico, when one can do in silico modeling of the whole flow of kinetic of the mycotoxin in the body by taking into account and incorporating physiological parameters. This is so-called PBTK, physiological based toxicokinetics. But therefore we need for each separate species right PB physiological parameters like organ blood flows, these Qs here. For instance, for liver, we need information on phase 1, phase 2 enzymes, both the expression, their activity we need. And in my opinion, for the moment, what is currently lacking really is knowledge about drug transporters, of transporter proteins. These APC transporters or SLCs are not well studied in several animal species, not in pigs. So food for thought and food for future, of course, here. And with this, I think, I don't know time exactly, but I think it's time to thank you for your online attention, and I'm happy to answer your question. I also want to thank the MyTox platform partners, and part of this work here in these young piglets was done within the MyCookie project, where Italian partners from ISPA, CNR are acknowledged for the coordination of the whole project. Thank you.
Thank you, Siska, for this great presentation. Marc, you have one question, I guess?
Yes, thanks, Siska, for this nice presentation. I was wondering if you have any hypothesis about the difference in the clearance between pigs and poultry, because I mean, it's so massive. And I was thinking, have you ever looked at serum albumin binding or maybe blood flow? Is it different for the blood flow between the renal blood flow between a poultry and a pig? Or can it be due to some difference in the kidney cells themselves? I mean, does anyone have looked?
So, sorry, Marc, can you repeat the first part of the question? There was a kind of heavy traffic here. I'm in the front of the road, sorry.
I mean, when you think about DON, for example, or anything is the same, but when you look at the difference in the clearance,
yeah,
we don't speak about the oral absorption. I speak really about the clearance when the molecules are inside the blood. Mm-hmm. It's massive. The difference is massive. In my knowledge, I don't think DON is interacting with serum albumin, so we can rule out normally.
No, we did. We checked the plasma protein binding to albumin or others, but in general, plasma proteins there was no binding.
Is it related to blood flow in the kidney that is higher, so more filtration, or is due to cell, for example, renal cells that are reabsorbed or not mycotoxin? I don't know because.
If you look to the kidney, you have to divide it in these 3 parts: the filtration and then the reabsorption. The filtration in a pig, it's quite similar as in human beings. An adult value is 120 ml per minute creatinine clearance.
And for the poultry?
But poultry, it's less actually.
Ah,
yeah.
So it's not logical.
It's not logical. And then for the reabsorption, I don't know, then you should calculate the ionized fraction or something in the.
Because if it's not retained in the blood, it means that it go to the kidney and then come back to the blood for pigs and not for poultry, for example, or something.
Mm-hmm. Yeah.
Is it possible to maintain the kidney, like, you know, to use ex vivo experiments with keeping the kidney and seeing if, I mean.
Yeah. I must say the broiler or the poultry research is always less well elaborated compared to pig research. So like to measure such type of things, but interesting, yeah.
Okay, thanks, Siska.
Veronika, would you like to ask your question now?
Yeah, thanks. So thanks, Siska. It's really cool to see all these differences. I really like that. I just wanted to ask you about some age-specific differences in the expression and activity of the glucuronosyl transferases. You've mentioned the cytochrome P4050, that there are differences, but have you checked also here in liver or intestine on this factor?
Yeah, we did not test it for this specific mycotoxins, but we tested for a drug, ibuprofen, an NSAID, an anti-inflammatory drug used in neonatology. People of university hospital wanted to know about this young piglet, neonatal piglet model. Could it serve to optimize the dose in really neonatal or even premature babies? And remarkably, this phase 2 glucuronidation is really rather rapid mature compared to CYP maturation, whereas CYP has some time needed to mature with age. It seems that for the phase 2 metabolism with glucuronidation, these young piglets are already full capacity of glucuronyl transferase being able to perform the glucuronidation. Yeah, there's a difference between CYPs and these phase 2 ones.
Okay, thanks. Thanks a lot.
Yeah, thank you.
I guess we'll have time for another very quick question. I would like to know, is your model only based in males, or do you also test females?
We usually test males and females, both equal numbers, to see these possible gender differences. But if we want to collect urine, I mentioned only males. Due to technical feasibility to stick these collection bags, you cannot do this in female pigs, because then you have contamination with the feces. It's too close by, and there we should catheterize, really put a catheter to the bladder or by direct puncture. But yeah, the alternative is, of course, going back in time and look for the metabolic cages. But at our university, the ethical committee is quite reluctant to acknowledge such type of studies when they are, even 1 day in a metabolic cage is already too long in their opinion. Yeah. So if you find alternatives ways, let me know for this female pig.
Okay. So I will going to pass on Veronika to the next speaker. Thank you very much, Siska.
It's my pleasure now to introduce Dr. Delphine Payros. She's working at the Institut de Recherche en Santé Digestive. You're giving me a hard time, huh, I have to say. So Delphine is working in the pathogenesis and commensalism of antibacterial in strong collaboration with biosynthesis and toxicity of mycotoxins teams from Toxalim in Toulouse. Her main field of expertise are the effects of co-exposure and food contaminants such as Deoxynivalenol and bacterial toxins, and the gastrointestinal pathophysiology and antimicrobial resistance. So Delphine, I'm pretty curious to hear about the role of DON in inflammatory bowel disease. The screen is yours.
Thank you. Good afternoon, and thank you to the organization for giving me the opportunity to present my work today. The subject of this study is to assess the role of the food contaminant deoxynivalenol, DON, on the inflammatory bowel diseases, so IBD, using an experimental animal model of chemical-induced colitis. So, as you know, the intestinal mucosa represents the first and most important barrier between the external environment and the host. And we can distinguish at first a physical barrier constituted by the intestinal and colonic epithelial cells and the mucus layer. Then an immunological barrier with the gut for gut-associated lymphoid tissue, and a microbiological barrier formed by the intestinal microbiota. The interaction between these different actors contributes to the physiology of the host. But there are also important players predisposing to the development of numerous gastrointestinal disorders, such as chronic inflammatory bowel disease. Mainly characterized by intestinal and systemic chronic inflammation, we can distinguish two main intestinal disorders: the ulcerative colitis, with a prevalence of 1 to 5,000 people, and with a site of the inflammation we continue only in the big intestine, from the Crohn's disease, with a prevalence of 1 to 800 people, and a discontinuous site of the inflammation all along the gastrointestinal tract. So these disorders are also often associated with a disruption of the intestinal barrier, and more precisely, the colonic barrier in the case of ulcerative colitis, for example, and a microbiota dysbiosis. Many factors are involved in the development of these pathologies, as genetic background, an inflammatory response excavated from the immune system, for example, an imbalance in Treg Th17 ratio, and a decrease of tolerogenic dendritic cells population. In the case of intestinal microbiota, a low complexity or diversity, or an increase of certain bacterial population as enterobacteria, could explain the role of intestinal microbiota in IBD. Finally, the last factor, the environment, one of the most diverse and most difficult to take into account, includes all the external factors to which we are exposed on a daily basis all along our life. The environment includes food contaminants, allergens, tobacco, alcohol, and among them, we wondered whether the contamination of food by mycotoxin, the most prevalent food contaminant, not involved in the pathogenicity of these disorders. Indeed, the gastrointestinal tract is also the privileged target for the action of mycotoxins, and among them, I got interesting in DON from the Trichothecene B family and mainly produced by Fusarium graminearum and Fusarium culmorum. DON is the most prevalent mycotoxin found in Europe, and it's commonly detected in cereal and grain, and in particular in the wheat in maize and in their byproducts. Therefore, the European Commission decided to limit DON level consumption in human food with some recommendations for animal feed. Actually, the toxicity of DON is well known, and there are consequences both in human and animal health. The main symptoms include abdominal pain, diarrhea, vomiting, and with lower doses for chronic contamination, it can decrease feed intake and body weight gain. Moreover, oral contamination with DON induces intestinal lesions, alters intestinal barrier integrity, and which very well described in the small intestine, and modulates immune system responses. So in this context, we can wonder what is the consequences of the exposure to DON-contaminated food on the development of colitis. So to do that, young rats were exposed to DON in their diet, and an acute colitis was induced with DSS for dextrans sodium sulfate, and we measured colitis onset, colitis severity, and inflammatory and immune responses. Here, the experimental protocol in detail. So male rats are fed with a control or DON-contaminated diet for 4 weeks. And during the last weeks of the experiment, we induce colitis by adding DSS in the drinking water. Weight gain and clinical symptoms were evaluated all the days during the DSS treatment. And at the end of the experiment, pro-inflammatory markers, morphological damage, the number of enterobacteria and immune responses were assessed. So as you can see on this graph, DSS here in the red curves induce a loss of body weight gain, and DON here exacerbates the decrease of the body weight gain. Concerning the clinical symptoms, DON in combination with DSS exacerbate the beginning of symptoms and aggravates colitis clinical symptoms characterized by diarrhea and blood in stools. At the end of the experiment, we analyzed at first the colon length, and as you can see on this graph, DON exacerbates the decrease of colon length in animals suffering from colitis. The analysis of microscopic damage showed increased neutrophil infiltration and epithelial damage in animals fed with DON-contaminated diets suffering from colitis, as compared with animals fed with a controlled diet. The study of pro-inflammatory markers in the colon revealed an increase of myeloperoxidase activity, an exacerbation of the production of interleukin-1 beta and CXCL1, reflecting greater neutrophil activity and infiltration in the colon. These results correlate with morphological analysis shown before. Then we looked at the number of enterobacteria in stools of animals suffering from or not from colitis. And as you can see on this graph, DSS-induced colitis also lead to fecal microbiota dysbiosis in favor of enterobacteria, and DON exacerbates this increase. To get further in the analysis of immune responses associated with the colitis, we analyzed the T lymphocyte population in the lamina propria. And at first, we assessed the percent of Th17 lymphocytes described to increase in IBD. As you can see on these graphs, the percent of Th17 increase in animals suffering from colitis, and a trend, not significant, but a trend is observed in animals fed with a DON-contaminated diet. And it's correlated with an increase of the secretion of interleukin-17 in animals suffering from colitis and fed with DON-contaminated diet after a CD3, CD28 stimulation. To analyze tolerogenic T lymphocyte response, we made the ratio of T regulatory cells on T helper cells. And as you can see on this graph, in animals suffering from colitis, DON decreases Treg T helper ratio, and it is correlated with a decrease in the interleukin-10 secretion, which is an anti-inflammatory cytokine. Finally, we looked at the dendritic cell populations, and as you can see on this panel, the both markers CD11BC and CD103 allowed us to differentiate procolitogenic dendritic cells from tolerogenic dendritic cells. And as you can see on these graphs, exposure to DON-contaminated diet increase the percent of colitogenic dendritic cells in the lamina propria of animals suffering from colitis as compared to animals fed with a controlled diet. And a decreasing trend can be observed in the same group for tolerogenic dendritic cells. In conclusion, the main results shown that combined exposure to DON and DSS significantly decreases the onset of colitis and exacerbates colitis symptoms. DON worsens the colitis induced by DSS through an increased production of pro-inflammatory markers in the colon, exacerbates dysbiosis in animals suffering from colitis in favor of enterobacteria, and modulates immune response with a decrease of Treg T helper ratio and a decrease of tolerogenic dendritic cells population in the lamina propria. So altogether, these results suggest that the presence of DON in the diet is a risk factor for inflammatory bowel disease and the exacerbation of the colitis in a rodent model. But to get further, we wonder if DON could be at the origin of IBD, for example, targeting the privileged site for these diseases, the colon. Indeed, few, only 1 or 2 studies analyze the direct effect of DON on the colon. But we found recently that rats exposed in their diet to low level of DON present many deleterious effects in the colon, with an hyperpermeability, a disruption of the tight junction protein, an increase of pro-inflammatory markers or apoptosis. And these results indicate that exposure to DON targets the colon, inducing a health-threatening breakdown of the colonic barrier. This highlights that oral exposure to DON is a risk factor in triggering IBD. These results are the subject of a publication submitted last week, so we keep our fingers crossed. Finally, to conclude, I want to thank all the people who participated in this study published last year in Archives of Toxicology, and thank you for your attention.
Thanks, Delphine, for this very nice talk. At the moment, I see that Laura has a question. Laura, please go ahead.
Yeah, thank you, Veronika. Thank you, Delphine. I have one question. I attended a seminar last year, and they were suggesting that inflammatory bowel diseases were probably related with a poor or modified antimicrobial response at the intestinal level that will actually change the sensitivity and the composition also of the microbiota, and also from the mucus that will be thinner in diseased people. Do you think that DON could be behind some of these effects?
In our study, we analyzed the antimicrobial peptides and stimulate some immune cells with E. coli peptides, and we don't see any differences. But we just measure the number of enterobacteria in this study. Maybe an analyze more precise of the microbiota be necessary. And a recent study from Vignal and I don't know the other authors published in the last year demonstrated that in a mice model exposed to DON in drinking water during 9 months, that there is an impact of total microbiota with an increase of proteobacteria, so and a decrease of firmicutes, I think. So maybe it's interesting to look more precisely the effect of DON on microbiota in our model, but we need to verify that. I don't know if I answer the question.
Yes, and do you think that that will be an effect of DON directly, or do you think that it changes the relationship?
I think DON induce an inflammatory state in the colon, and so in favor of the proliferation of certain bacteria as enterobacteria or E. coli, and it's one of the factors deleterious, bad factor for privileged the appearance of the kind of disorders.
Okay, thank you very much, Delphine.
Thank you, Laura.
Yeah, so Laura stole my question. I have to say I wanted to ask the same. But one last question from my side, as I don't see any from the audience at the moment. Are you also going to test other mycotoxins in this regard? So maybe other trichotethanes, and do you expect more or less a similar mode of action?
Also, I understand I heard the beginning of the question. So if you look at other mycotoxin in this model, it's this question.
Yes.
Yes. So we looked at another mycotoxin derived from DON. It's the D-epoxy deoxynivalenol, a less toxic form. Less toxic form of DON, and we have exactly the same results as than in the control group. So DON 1 did not exacerbate the clinical symptoms, body weight loss than DON. But we don't. We didn't test another mycotoxin.
Thanks for that. I think Siska has a question on your talk.
Yeah, thanks, Delphine. Just a short one. Would it be an option to find some cohorts, groups in hospitals, let's say, of some leftover plasmas of these people to check for the biomarkers? Yeah.
Yes, I think it's an idea, because a lot of research is necessary to confirm and to validate these results, these experimental results, and maybe look at patients from Crohn's disease or ulcerative colitis, look at the enterobacteria, and does the DON or the other mycotoxin in their stool or in their urine or plasma. It's very interesting, and it's not already done, so it's a good idea, but we don't check this yet.
Okay, thanks you.
Thank you.
So then thanks again, Delphine. I think there's a lot more research still needed or interesting to do. And with this, I hand over again to Laura for our last presentation.
Yeah, well, I have the pleasure now to introduce you to Dr. Marc Maresca, who is currently a researcher at the Aix-Marseille University. He was awarded his PhD in biochemistry from the Université Paul Cézanne in France in 2003, where he worked on toxins causing intestinal pathologies, including mycotoxins. In 2003, he moved to England to work on the bacteria enteropathogenic E. coli in the laboratory of Brendan Kenny, and then he moved back to France to continue his work on mycotoxins, mainly deoxynivalenol, studying his effects on human health. He's currently working at the Aix-Marseille University, and his research aims, in addition to study mycotoxin effects on health, is to identify and develop new molecules with antimicrobial properties, be it natural, synthetic, and bio-inspired, and including the ones that are produced by microorganisms. Marc, now is your turn.
Thanks a lot, Laura. Hi, everyone. So I'm Marc Maresca, and I would like to speak about another aspect of mycotoxin with the last presentation. All the other speakers present how the mycotoxin can affect plants, animals, or humans badly, meaning that they cause toxicity or they cause infection in plants. And I would like to present you another aspect of mycotoxin, the potential use as therapeutic drugs. And for that, I will take a particular example that are fungal cyclodepsipeptides. To be sure about the time, I will be fast in the beginning because it's general introduction, and then I will go for the experimental part to spend more time on. As you certainly know, fungal metabolites, secondary metabolites from fungi, are most of the time produced as an answer to the ecology of the fungi. It could be to resist, for example, UV lights, to interact or to compete with bacteria, and they could play a role during the fungi development or biology. And when you look at this metabolite in terms of chemical class or chemical family, some of them are derivative of acetyl coenzyme A or derivative of this, and leading to the polyketide family of metabolites. Also, mevalonate that leads to terpenes, including mycotoxins. And another type of metabolite that will be presented today are derivative of amino acid and related to peptides. And here you can see some of very well-known and good example of metabolites that can be used as drug in medicine. We will talk about this later, but levetiracetam is used in medicine, cyclosporin too, penicillin is also used as a drug for treating disease. So as I say, secondary metabolite from fungi can be very dangerous, deleterious. And the best example for that is mycotoxin, as we already see it with different speakers. And on another hand, there is quite a lot of drugs used in medicine that are secondary fungal metabolite. Penicillin is one of them, used as antibiotics. Paxiclatel used for treating cancer. Levetiracetam to decrease cholesterol, cyclosporin as used as immunosuppressant, zearalenone that can be used as a growth promoter, and so on. And when you think about this, mycotoxin certainly caused thousands of deaths in humankind from other history. But in the meantime, good metabolites save millions of humans. And what is important with all these molecules and with the drugs in general is it's about the dose. It's all about dose. And what makes a good or bad action of a molecule will depend on the dose. And this is very old because it was already wrote and said by Paracelsus. And what is making the poison is the dose itself. And when you extrapolate this to experiments in lab, all this about good and bad effect of a drug depending on the dose is what we call therapeutic index, or TI. And the TI is calculated, as you can see here, by dividing the effect on the bad effect, toxic effect, and taking into account also the positive effect, so the therapeutic effect, sorry. And you have four kinds of scenario with the TI. The first one is the best one, is what we expect for medical drugs. So here you can see that the beneficial effect is obtained at very low dose compared to the toxic effect. And some molecule can reach this not so good situation, but still can be used as medicine, is when the beneficial effect is very close from the toxic one. The last two scenarios are the worst one. No therapeutic margin means that, in fact, the therapeutic index, the beneficial effect and the toxic effect are observed at the same dose. And the worst of the worst is when the toxic effect appears before the beneficial one, if there is any. And this allows you to obtain TI value ranging from 10 to 100, 1000. It's very good. TI ranging from 1 to 10, not so good. TI equal 1 and TI lower than 1 that are the worst case scenario. And there's a difference between the safe drugs and a toxic drug. Some example, penicillin and levetiracetam are very well tolerated by humans with a TI higher than 100, 1000. Cyclosporin and paclitaxel, have TI lower than 10. So they are not very good. But I mean, when you have no choice, such as cancer, you prefer to have this kind of TI than nothing. And most of the mycotoxins are here with a TI lower than 1. So they are not really drugs. They are more likely toxin. What about cyclodepsipeptide from fungi? So just for you to know about this kind of molecule, there are peptides produced by fungi, but also by plants and animals, bacteria as well, sorry. And actually, this is a quite old review about this because every month, every year, you have new cyclodepsipeptide found. But in 2018, it was already reported in this review more than 300 fungal cyclodepsipeptides. And this review also summarized their potential therapeutic application. And one of the ones that I will present today is their potential antimicrobial effect. What are cyclodepsipeptides? So normally a peptide is produced by a ribosome, and this is a classical way to produce a peptide or protein. So you have the DNA coding for the RNA, and the RNA is converted to protein by translation. This is a classical way to produce a protein or a peptide. Cyclodepsipeptides are different the way that they are not produced by ribosome. So they are what we call non-ribosomal synthesis production. And the enzyme causing this production are synthetases that are SAM dependent. So SAM for S-adenosine methionine. And here it's drawing, it's a case of the eniacin that are imaging mycotoxin, cyclodepsipeptide also, produced by this kind of synthetase. And here you can see that the enzyme, the synthetase, use two types of amino acid and make like cyclic peptides that can have 2 to 13 amino acids long and giving a large number of molecules with different size and composition, of course, in sequence. What is important is when you look at the literature, you don't find so much therapeutic index determination of the cyclodepsipeptide. Most of the paper describe their activity, but they don't care really about the toxicity. And it's very hard to know if they can be used really or not as drugs. So this was the starting point for our experiments and what I'm going to present you today. And so we decided to look at the cyclodepsipeptide from fungi, taking eniacin and bovericin as the first example. I will not take too long on this, but they are produced by different fungi, including Fusarium, but also different one. And they are what we call cyclodepsipeptides, so 6 amino acids. And you can see here that you have 3 different positions, either containing a sec-butyl, an isopropyl, or phenylmethyl group. And by changing R1, R2, R3, you obtain eniacin A, A1, B, B1, and bovericin. The thing is, when you look at the literature, as for other cyclodepsipeptides, you can find things that they say that they are bad. So eniacin and bovericin are part of the emerging mycotoxin, meaning that they have no regulation for this kind of molecule. They are present in food. They can contaminate grain, cereal, but also meat and fruits. And the thing is, when you look at the literature, you can find toxic effect at micromolar dose. In animals, the lethal effect, 50% lethal effect, is around 10 milligram per kilogram per IP treatment, so injection, intraperitoneal injection. When you give them orally, you go to 100 milligram per kilogram of body weight, in mouse at least. And in the meantime, when you look at the whole literature, you can find that eniacin and bovericin were described as able to inhibit and kill bacteria, but also to possess antiparasitic, anti-cancer, and anti-inflammatory activity. Depending on the paper, you will find some ranging active concentration around 1 micromolar, 10 micromolar. Okay, and this leads, in fact, to the use of eniacin already as therapeutic drugs. In fact, it was a combination of eniacin called fusafugin, that was used to treat airway infection topically. And this molecule was very well used in a different country, but nowadays is withdrawn from a lot of countries due to noticed toxic side effect. And we will explain this later on. The objective of the study was the following. First, to understand how the eniacin and bovericin are able to kill bacteria. And second, in the same study, to look at their antimicrobial activity and toxicity on the same study with the same molecule to be able to determine the toxic and the therapeutic index. And this was published already in Toxins last year, 2 years ago, sorry. First, we test them as antimicrobial, and we found something that was already described, meaning that they are mostly active on gram-positive bacteria, with antibacterial effects ranging from 1.5 to 100 micromolar. They also show activity on Mycobacterium and against some fungi, particularly Candida, but also Fusarium graminearum, but not other fungi we tested. Interesting also, we found that eniacin B was almost non-active on any of the strains tested except Clostridium perfringens and Candida. We looked at the mechanism that was not determined in the literature, and we looked at first, are these molecules able to compromise the bacterial membrane? We have to know that antimicrobial peptide most of the time target the membrane of bacteria to kill them. So we look at the permeabilization of the bacterial membrane by eniacin and boverecin. And we found that, indeed, these peptides are able to permeabilize the bacteria. But this permeabilization occurs at very, very high doses compared to the antimicrobial concentration. What we call MIC value is for minimal inhibitory concentration in bacteria. They are ranging here between, I would say, 3 to 12 micromolar. And you can see here that on this bacteria, the permeabilization occurs only at 100 micromolar. Same for the depolarization of the membrane. You can see that also eniacin and boverecin can cause some depolarization at the MIC value. For example, eniacin B is not causing any depolarization at this concentration, or very weak. Meaning that depolarization can be part of the mechanism of action of this molecule, but it could not explain all. And so we decided to move to another approach to study the mechanism of action of the eniacin and boverecin by using inhibition of macromolecule synthesis assay. So when you want to look at inhibitor of synthesis in bacteria, you can either use radioactivity, and I will present you some data about that later. But in our lab, we have the chance to get Clarisse Roblin, that she was in PhD last year, or 2 years ago, I don't remember. And she brings with her a very nice technique that is a microscopic technique to study, evaluate, and determine how a molecule can affect macromolecule synthesis in bacteria. So to be simple, what the technique is this: you incubate the bacteria with non-antibiotics targeting either cell wall, DNA, RNA, or protein synthesis, so amoxicillin, gemifloxacin, rifampicin, and tetracycline. They are well known to affect either cell wall, DNA, RNA, or protein. Then you stain the bacteria with the membrane probe that stain membrane in red and a probe that stains the DNA in blue. And then you look at the phenotype. And you can see that the phenotypes are different. For example, DNA synthesis inhibition makes the bacteria very, very long. With the protein inhibition molecule, you can see that DNA is getting like hypercondensated in some dot inside the bacteria with no elongation of the bacteria. I mean, each kind of antibiotics with a non-inhibitory effect give a different phenotype on the bacteria. And then you compare with your molecule that you don't know about the mechanism of action. So here is eniacin A, eniacin A1, B, B1, and boverecin. And if you zoom in, you will see that eniacin A and A1 makes the DNA looking like a, like a worm inside the bacteria. You see here? And this is also happening with rifampicin, an RNA inhibitor. For eniacin B, B1, and boverecin, sorry, you have this phenotype looking very close to the tetracycline phenotype, suggesting that eniacin B, B1, and boverecin are able to inhibit protein synthesis. The important question about the eniacin was not really about the mechanism of action. It was more likely about the therapeutic index. So we look at different human cells to determine the toxicity of eniacin and boverecin. First, we did a very simple assay that is looking at the hemolysis effect of this molecule. And you can see here that they are not so much hemolytic. They can cause some hemolysis, but at very high dose, very high compared to the antibacterial effect. So if you consider the hemolysis, they are safe, with a TI ranging from 45 to more than 300. Problem is when you look at nucleated cells, so normal cells, and here is different. So we test different cell line, airway, sorry, intestinal, dermal, liver, and endothelial cells. And you can see here that, I mean, they are very sensitive to eniacin, much more than the red blood cells, with an IC50 on cell variability ranging from 1.1 to 54 micromolar. And this is very bad. This is very bad because here is what we do with Hamza Holek, that is my former PhD, is plotting on the same graph the effect, the beneficial effect, so the killing of the bacteria versus the killing of human cells. And here you can see here clearly that they are not good at all. So for example, on Eniacin A, A1, B1, and Bovericin on Staph aureus, you can see here that the TI is ranging from 0.5 to 1. Eniacin B for Staph aureus is even worse. No effect at all on staph, but very toxic compared on the airway cells. The only target of Eniacin B was Clostridium perfringens, an intestinal pathogen. So we look and compare with CACO2 cells. And here again, the TI is lower than 1. So all this clearly show that compared to classical antibiotics used in medicine, Eniacin and Bovericin are very bad, very, very bad. And it completely explains why this kind of molecule were withdrawn from the market nowadays, because they are toxic, clearly, to human cell more than from bacteria. So now what I'm going to present you fast, because I'm quite short in time, is what we get with another example of cyclodepsipeptide from fungi. I will not give you the name because we are still not publishing the data, because we are compiling a lot of results, because it's a very, very interesting and very nice study. So I can tell you the name of the molecule. It's secret, but I can tell you about the experiments and what we get with this one. So we compare the new peptides with Eniacin and Bovericin. First of all, the new peptide that I will call peptide X is very, very, very selective, meaning that when we looked at the activity of the peptide against a broad spectrum of bacteria, it was only active on Clostridium perfringens. I only show you some examples here, but we tested more than 40 bacteria and fungi, 40. And none of them were sensitive except Clostridium perfringens. Another good news was that we tested a clinical strain from animal and human of Clostridium perfringens, and we confirmed that it was very selective of that Clostridium perfringens. So peptide X looked like Eniacin B, a very, very narrow selectivity against Clostridium perfringens. It was also confirmed with a collaboration with Adiceo group, this is a company, because they use a chicken cecal content in which they introduce Clostridium perfringens, treat the cecal content with our molecule, and the molecule only kills perfringens in the middle of billions of bacteria. All the rest of the bacteria were not affected. About the mechanism of action, we started with the microscopy, and like Eniacin B, we see this kind of dot inside the bacteria, and suggesting that the molecule is inhibiting protein. So as we are very excited by the study, we went to Toulouse, Université Paul Sabatier, to do a study with radioactivity. And we confirm that the protein, the peptide X, sorry, is able to inhibit protein synthesis and to a less extent DNA synthesis. So peptide X is different from Eniacin A, A1, and Bovericin, but more likely act like Eniacin B, B1, or Bovericin in terms of mechanism of action. Okay, so we were not so happy because we were not sure about the toxicity of peptide X. So we went to the therapeutic index determination, and here is result. It's very nice. So as we target perfringens, we decided to use intestinal cell model, but after that, we confirmed that the molecule is not toxic to many other human cells. And you can see here, very active on perfringens and no toxicity at all, up to 100 micromolar of peptide on CACO2. We don't went higher because we can't due to the protein peptide amount that we have in the lab, but up to 100 micromolar is not toxic at all, meaning that the TI is at least up to 66. And when you compare this to Eniacin B, that having a TI of 0.3, you have no comparison for the safety. Conclusion was peptide X is much, much safer than Eniacin and can be used potentially in vivo. So we decided to go further and to continue the characterization of the molecule as potential antibiotics to use in human and animals. So you have to understand that, and I'm pretty sure that you know, but antibiotics most of the time select resistant strain, and this is a very bad point about conventional antibiotics. So here's an induction resistant test in Clostridium perfringens. You can see here that metronidazole, that is a conventional antibiotics, as expected, cause resistance over time. So if you incubate Clostridium perfringens and expose it to metronidazole during times, we see that the MIC, so the sensitivity of the bacteria to the conventional antibiotics, is going low because the MIC going up. With peptide X, no change. So peptide X is not causing an induction of resistant strain over 30 days. Next, we wanted to know if it's working in vivo, because when you want to have a drug to use in animals or human, you have to show that it's working in vivo. As we don't have at that time an in vivo oral assay for Clostridium perfringens infection, we did a simple one that is intraperitoneal infection. And you can see here that normally when you infect intraperitoneally mice with CP, Clostridium perfringens, they die in 6 hours. Vancomycin, that is used to protect the mouse as a conventional antibiotics, working perfectly, saving life of the mouse over time. And with the peptide X, depending on the dose, we see different results. With a low dose, we have some protection. A medium dose, the protection is still on, but with a high dose of peptide, we were hoping to have the same result than with vancomycin, but unfortunately, at high dose, peptide X kills the mouse in like 3 hours. But this is just like eniacin. If you inject eniacin at 10 milligrams per kilogram IP, the mouse dies. So it's even less toxic than eniacin, but it's still toxic in an IEP model. We also confirmed that the bacterial count was lower here. So really, in vivo, peptide X is able to protect partially, at least, the animal from a perfringens infection, a systemic one. So nowadays, what we have a drug potentially used for medicine. We have to make it better because injected IP is killing the animals, so potentially will do the same to human. But we can imagine modifying it or using vectorization to make it less toxic. Another important issue is we're going to do some oral infection with CP and to treat with peptide X orally. The thing is, we believe that orally the peptide will be less toxic, like for eniacin. I would like to thank all the people that perform all this study, eniacin and peptide X. So Hamza Olek is now a postdoc in Compiègne, but he was in the lab at that time. So all the people from my lab in Marseille, people from Toulouse University, the LMGM lab, some people co-supervising Hamza with us in Lebanon, Michel Fons, that is in Marseille too, Catijano that perform some tests on clinical strain, Adiceo that performs the fecal content assay, and Cédric Jacqueline in Nantes that performs the in vivo data and assay. I would like to thank Aix-Marseille Université, CNRS, and the INRAE that give us money to perform experiment through the grant emerging mycotoxins. Professor J. Fox for giving me the opportunity to talk and to present toxin and DPI the same. And of course, I would like to thank all participants for your attention. Thank you.
Thanks, Marc, for your very nice presentation. We don't have questions for the moment, but I have one. Do you have an idea why it's only useful in Clostridium perfringens and not other Clostridia?
This is the major issue with this study. So actually, each time we delay the publication because each time we believe that we can make it better and to target a bigger journal. Problem with the molecular target of antibiotics is usually you find the target by inducing a resistance. So you take the bacteria, induce resistance, and then sequence the resistant strain compared to the naive one, and the mutation is in the target most of the time. Okay. Problem is here that it does not cause resistance first, and second, the peptide it's impossible to label it. Same. If you can label your molecule, you can follow it, meaning that you can use it for fishing the target inside bacterial supernatant or bacterial extract. You can use your labeled drug to bind to protein or something when you do an extraction of protein. So we can label the molecule, except by radioactivity, but it will be hard and time consuming and not very good.
Kilos of.
And we can't have access to a mutant resistant to the molecule because it's not causing resistance. So my opinion is, for sure, it's very specific of perfringens because it's not active on other Clostridium. I didn't show it, but on the 40 strains that we test, some of them are Clostridium. So difficile, Nexile, Cocoides, I don't remember the last one. So we test a lot of different Clostridium just because we wanted to know if it was directed against all Clostridium or not. Unfortunately, it's not active on difficile because if it was active on difficile, it would be even better. It's very, very specific of perfringens. I have to say that when we use, when Adiceo performed the assay on cecal content, they found another target. So we have to confirm this using an ATCC strain because Adiceo have 60S RNA sequence to give us a second target. But nowadays, I have to buy the strain and to perform an MIC assay to be sure about that. But it suggests that a second bacteria is sensitive to this molecule, but only 2 on millions, because cecal content is millions of bacteria. So why these 2 bacteria are sensitive and not the rest? No idea. No idea at all. I would say that they certainly bind to something that is different from Clostridial perfringens to another Clostridium. Unlikely. Another option is potentially that the Clostridium perfringens are able to activate the peptides, modify it to an active form. But we try this. So we incubate the molecule with perfringens and try to see after incubation, the molecule become active on other bacteria. It's not the case. So sorry, I don't know.
Okay. I hope you will know soon.
I think we will never know.
Okay. Well, if nobody else has more questions.
I had a small one, if I may, to close. Marc, sometimes in literature it's mentioned that these eniatines and boviricin have properties like the ionophoric antibiotics, the coccidiostats.
Yes.
Did you look for this type of action, the Emeria?
You have it. Yeah, we found the same. So I'm sorry because I think I was too fast. When you look at the graph here is depolarization of the membrane. So when you get depolarization, meaning that you have a ionophore effect because it's not linked to a permeabilization, a full permeabilization. So here, what you see here is ionophoric effect, at least of eniacin A, A1, B1, and to a less extent, boviricin and eniacin B. But yes, and they are reported to be ionophoric. But again, it's all the problem is the dose. If I use 100 micromolar, I would say that they are performing, but this is not the active dose because the active dose is here. So I think they are ionophoric, definitely, but I think they may affect also protein synthesis and DNA synthesis independently of the ionophoric effect.
Okay.
Okay, well, so yeah, there is some question from Mathieu Souris. Maybe he wants to make the question himself.
Hi, Marc.
Hello, Mathieu.
I want you to ask you, what if you perform random mutagenesis on Clostridium perfringens and then you test proteins on it for maybe identify the target of this toxin?
It's a very good point. It's a very good suggestion. We thought about it too, but it's a lot of work without be sure that it works. And second, I'm not sure also that we necessarily found the target because you can imagine that you mutate and upregulate an efflux pump, and you will see a decrease in MIC without any without being linked to the target itself. So we thought about this. But we have to find someone that have an aerobic chamber that agree to put some mutagenic molecule to cause and increase the mutation rate and then to give us all the mutant to screen for the MIC. I think this could be like the following of the first publication, but nowadays Hamza and I want to publish the study, so I will not have to put any more peptide X, and we may continue with doing what you suggest. We thought about this. This is a very good point, a very good suggestion, but we need to have either to do ourselves, because we have the anaerobic chamber, but using mutagenic drugs to make increasing the rate of mutation in bacteria, I'm not very happy with this.
Yeah. Okay.
You can come in the lab if you want.
Okay, thank you.
So if nobody has more questions for Marc?
I really would like to thank all the chair, all the speaker. It was a nice time. It was a real nice time to be with you. And I hope the participant learns a lot about the mycotoxin, the different aspect of the mycotoxin. And I hope we will meet again during some kind of webinar or conferences, and to promote the mycotoxin either on the toxicity part or on their use as drugs. And thanks a lot to all of you. Charlotte, you want to?
Yeah, I just wanted to thank all of you, same really, for your time and your efforts and your research, very interesting. And yes, I hope we are able to meet again, maybe in person, would be nice. Have a nice day, morning, evening, afternoon, wherever you are. And bye-bye, everyone.